Hi, everyone. Thank you for attending our PSF Major Donor Virtual Update, and welcome. I'm Pacific Salmon Foundation Vice President for Salmon, Jason Wong. I'd like to begin by acknowledging the Coast Salish people, Squamish, Tsleil-Waututh, and Musqueam peoples, on whose traditional territory we're privileged to gather today. And we recognize that you are all coming to us from the many traditional territories across and beyond BC. PSF typically hosts major donor events to connect with those loyal donors who contribute annually to our work. It's an opportunity for us to say thank you, to update you on our activities, and to engage in a topical discussion on wild salmon. But due to COVID-19 restrictions, we've been adapting. Back in February, we held our first virtual major donor event, and we're continuing to adapt to stay connected with you and to bring you interesting and informative content about wild salmon and the work of PSF. Tonight, we're trying something new again. We've expanded our reach a little further, since one benefit of shifting to an online format is that we can all connect from different regions, whether we're on the mainland, Vancouver Island, or up north. Corey Matheson and I are here in studio, and we have Dr. Brian Riddle and Dr. Isabel Pearsall joining us from their home offices. Brian and Isabel are going to provide us with an overview of the amazing work that continues to be accomplished through the Salish Sea Marine Survival Program, discuss how the work has been contributing to applied actions for salmon, and talk about the work that we're continuing with, building off the legacy of the SSMSP. PSF recently published the Canadian Program Summaries Report focused on this material, which you all have access to and which will be publicly shared for the entire salmon community. Hopefully you've had a chance to read through the document. It's a fascinating breakdown of many key elements of the SSMSP. And if you haven't had a chance to yet, you'll definitely want to dive in following tonight's presentation. So first off tonight, we're going to have a 25-minute update on key findings and next steps related to the Salish Sea Marine Survival Project and Program. And then we will move into some time for questions and answers, where we can ask Brian and Isabel questions from you, the audience. I'll act as your moderator tonight, and Corey will be managing the Slido. And before we get started with the presentations, I'll turn it to Corey to explain how you can send us questions for Brian and Isabel. So over to you, Corey.
Right on. Well, thanks, Jason. And as Jason mentioned, I'm going to be keeping an eye on all the questions that you ask here tonight. If you do have a question, you can enter it at any time, starting now, and you can do so through the Slido box, just on the right-hand side of the web page you're viewing. Simply click your cursor where it says type your question, enter your name below that, and then go ahead and press send. I should note we've got a probably larger than usual audience here tonight, so if we don't get around to answering all of your questions on the live part of tonight's show, we will be sure to follow up with you after the show, as long as you make sure to include your name when you submit your question. For you techies, you can also scan this QR code that should be on your screen right now and enter your question via your mobile device. Don't be shy in getting your questions in. I'm going to get back to my laptop now to start watching those questions and comments roll in. Jason, back to you.
Well, thanks, Corey. This live stream, as Corey indicated, is being watched by friends and supporters province-wide and will be recorded to share with those who couldn't tune in tonight. Our donors are key to our success. Your support enables PSF to be a leading voice for priorities and action for our wild salmon, and we're grateful for your ongoing support. So just before we turn to the presentations, I'd like to mention the pivotal role of Brian Riddle in envisioning and undertaking the Salish Sea Marine Survival Program beginning around a decade ago. Brian's experience, knowledge, and vision for what was needed and what was possible was one of the essential ingredients behind this successful program. And I wanted to make sure that this was acknowledged as we begin this evening. And now, let's turn to the presentations from Brian and Isabel.
Well, good evening, everybody. Wonderful to meet with you again, even if it is virtual. And tonight we have the pleasure of talking about the Salish Sea Marine Survival Program as it is now finishing up. And we've provided all of you, I hope, with the summaries that Isabel was largely responsible for, but we've prepared over the last couple of years. So we're going to highlight our program for you, and we're going to spare a lot of the details. No introduction to the Salish Sea Marine Survival Program would really be adequate without acknowledging the support from the board of directors and especially to you donors. It has been spectacular support. The logos listed on this slide are those donors who contributed over $50,000, and there are many, many others who supported with financial contribution, time, in-kind support, and just moral support generally. We thank each and every one of you for your contribution to this effort. What we're doing about it is going to be a series of slides by Isabel Pearsall and myself explaining the basic results and where we're going in the future. We want to start with just a general overview of the primary objectives of the research. You're probably all familiar with the principal goal was really to look at the factors that principally determine variation from year to year in the production of salmon in the Salish Sea. But that was the international objective, and the Pacific Salmon Foundation set a number of sort of sub-objectives for the foundation itself. Working with communities, sustaining future fisheries. Many of these benefits will accumulate over time. But one that we will come back to in the talk is to provide a long-term foundation for monitoring the health of the Salish Sea and our Pacific salmon. This is something that was badly neglected and led to many years of delay between when events occurred and when we finally did something about it. Let's start with the original infographic describing the scope of the Salish Sea Marine Survival Program. And many of you may recall that I described an ecosystem approach as doing everything, everywhere, all at the same time. Obviously a challenging task, but I'm very pleased to report that we delivered on all the topics in this slide, with the exception of the contaminants, and that was really addressed more fully by our U.S. partners. As in the program summary booklet, tonight we will summarize the results in just 6 research themes, and we will spare you all the apparently painful details that we've discovered along the way. Oceanography is one word that encompasses all aspects of the bottom-up process that annually determines the food for salmon and other fishes. These programs are the basis for understanding the variability in salmon production over time and space. Has the biological productivity of the Salish Sea changed in recent years? And how can we assess something as complex and dynamic as the Salish Sea? How to tackle this was a major success of the Salish Sea Marine Survival Program. The use of community volunteers, our citizen scientists, throughout the Canadian Salish Sea enabled us to assess the entire Strait of Georgia within 1 day with sampling intensities never before accomplished up to the level of zooplankton, and we relied on DFO science to fill in that portion of the science. Has the Salish Sea productivity decreased over time? The answer is no. Our analysis indicate that change in annual production hasn't changed, but there is evidence of change in the timing of events that we refer to as a match-mismatch issue. We can't eliminate that possibility, but we can continue to monitor it with our Salish Sea program in the future. And with respect to PSF's objective for long-term monitoring, the development and continuation of the citizen science oceanographic program will provide the long-term monitoring needed to address the changes that we'll expect to see in the future. Development around estuaries and neglect of estuaries and nearshore habitat is widespread and is a significant problem in the Salish Sea, but it's one that we can't address. Salish Sea Marine Survival Program studies of marine plants, again through many of our community scientists, and the estuary and shoreline restoration indicates that we can restore these critical habitats, but it's going to be a large and long task to undertake. We expect restoration of these habitats and marine plants will have a significant benefit to Pacific salmon and other components of the salmon ecosystem throughout the Salish Sea. More complex, healthy marine plant communities provide better shelter, food, and diversity for many fishes. And an abundant and diverse fish community is likely to reduce predation on juvenile salmon. While there's lots to do, it is all doable and we can undertake it. Research in the Cowichan River was a huge component of the Canadian Salish Sea Marine Survival Program, and in many aspects, it demonstrated the essential issue of river flow and survival for juvenile salmon. It enabled studies of predation in the river and in the estuary. It provided the first direct estimates of predation on juvenile salmon using DNA analyses of the poop. It enabled the most direct comparison of hatchery and wild Chinook ever undertaken, and it demonstrated the unique value of passive integrated transponder tags and the development of micro-trolling as a way of tagging and sampling juvenile salmon. This program has stimulated a much bigger bottleneck study to production that Isabel is going to describe for you in the second half of the presentation. In salmon, we refer to a bottleneck as a significant limitation to future production. In this stylized curve of survival over time, the steep descending left curve, accounting for 80% of the loss, would be considered a bottleneck. We also describe this as a critical period in salmon biology. But during the Salish Sea Marine Survival Program, the application of pit tags throughout the early marine survival of Chinook allowed us for the first time to partition the mortality during the early marine period, and it demonstrated the continued mortality of salmon after leaving the nearshore, possibly and likely during their first winter at sea. Plus, our pit tag studies clearly showed that the survival of wild Chinook was twice that of the hatchery Chinook in the Cowichan River. Studies of salmon while they rear in the Salish Sea was a continuation of DFO's science programs originally developed by Dr. Richard Beamish, but with much expanded sampling for populations of origin and health conditions of the wild salmon. And this certainly paid off. Coho and Chinook juveniles use the Strait of Georgia differently. Coho salmon are much more of a generalist, with many populations intermixed throughout the strait, whereas specific Chinook salmon populations were shown to annually utilize specific areas of the strait. These observations corroborated results determined statistically using code-award tags of hatchery fish. Plus, the surveys clearly demonstrated that the summer Chinook, particularly Thompson River, enter the Strait of Georgia in the late summer, after the summer bloom and after all the fall Chinook have either died or have emigrated from the region. These analyses and surveys are being continued by DFO Science and will continue to provide critical information. Our work, simply stated, was foundational. The innovation of seal beanies, the development of genomic tools to identify species in the poop, and the three-dimensional tags to study feeding behavior have changed how researchers consider the issue of seal predation. No one expected the cumulative predation rates estimated on juvenile Chinook and Coho. Of course, for much of this development, we have to acknowledge Dr. Andrew Trice at UBC that had spoken to the major donor circle before. Unfortunately, our success has also led to our involvement in issues of how to manage seal predation in the future. Daily predation rates were only about 6% per day on the big Qualicum Coho salmon, and even lower for small Chinooks. But given the population of harbor seals being around 40,000 in the Strait of Georgia alone, the estimated loss of Chinook and Coho salmon was between 30 and 40% in total. Predation rates, though, may be reduced through habitat restoration, increased forage fish abundances, and management of log booms that we still allow in our estuaries. The SSMSP also touched on the topic of predation by birds, but I think that this is a bit of a gap in our information right now. And there is the broader consideration of the other marine mammal species and impacts on adult salmon as they return to spawn. Without exaggeration, these studies fundamentally changed how we can study fish health and disease. But as you are likely all aware, they've also drawn us fully into the debate about open net pen salmon aquaculture and effects on wild Pacific salmon. For example, the next slide is a heat map of individual fish and pathogens, providing over 7,500 observations in a single assay. In the end, we assessed 60 pathogens in 31,000 salmon, resulting in 3.5 million observations. Applied results from this research are difficult to encapsulate briefly, but include 15 new viruses discovered, cumulative evidence that open net pen salmon aquaculture presents more than minimal harm to Pacific salmon, and provided new methodologies to study salmon in hatcheries, cumulative effects of the environment, and can be applied to future studies of climate change and salmon. It's not an exaggeration to conclude that these studies may revolutionize our ability to study the health of many species in nature. To conclude this work, I have to acknowledge the scientific insights of Dr. Christy Miller, Emiliano DeSicchio, and several postdocs, including Andrew Bateman, who is now part of the PSF staff. As a final comment on the Salish Sea Marine Survival Program, the collaboration between partners from many organizations in Canada and the United States speaks to the scientific credibility of this endeavor and the efforts of Dr. Isabel Pearsall as our program manager, and of course to the donors that trusted us and enabled this research from the beginning. Many, many thanks for your support. But our efforts are going to continue, as Dr. Isabel Pearsall will now present. So it's my pleasure at this time to introduce you to Dr. Isabel Pearsall, our Director of Marine Studies at the Pacific Salmon Foundation.
Thanks, Brian. Good evening, everyone. Thank you very much for being here tonight. My name is Isabel Pearsall, and I've had the pleasure of working alongside Dr. Riddle over the past decade, managing the Salish Sea Marine Survival Project, and more recently developing the current legacy programs that have been designed to build on our results from SSMSP. We've now developed a broad program titled the PSF Marine Science Program, and today I'm going to let you know about some of the projects that we are working on. As Brian mentioned, one of the SSMSP goals was to provide a foundation for long-term monitoring, and moving forward we are funding several citizen science programs aimed to increase our understanding of the Strait of Georgia ecosystem. Here we're showing a photo of a group out of VIU that work with us to train citizen scientists around the Strait to sample beaches for forage fish embryos, focused primarily on sand lance and surf smelt. By identifying beaches that are positive for forage fish, a vital component of the food chain, including for salmon, we can work with other groups such as WWF Canada to find solutions to protect these habitats and beaches. Brian also mentioned our Citizen Science Oceanography program. This program is highly regarded and is continuing thanks to the support of our funders. We've been very lucky to have great retention of many of our citizen scientists who have been with us since the beginning in 2015. The map on this slide shows the current sampling regime, and you can see the excellent coverage throughout the Strait of Georgia. And there's also a photo of Svetlana Esenkulova. She's one of our PSF staff, works with us on this program, and she's holding one of the key instruments, a CTD, which measures water properties. Now we're in our 7th year, which is fantastic, and providing data that are useful now to start looking at some of these longer term trends. And we work closely with Dr. Rich Pavlovich, a UBC oceanographer, and he has created an atlas of the Strait of Georgia oceanography, which is now digitally available on PSF's Strait of Georgia Data Center. Rich has let us know that this data set should be the new standard for the Strait of Georgia, and that much of the research that is ongoing, particularly in his lab, would not be feasible without these very detailed data sets. Another citizen science program that we help fund is called the Coho and Chinook Adult Diet Study, and this was designed and developed at UVic by Dr. Will Dogood, who is currently with us as a postdoc. This program uses the salmon themselves to sample the ecosystem. So recreational anglers cut out salmon stomachs when they catch a fish, and then they freeze those stomachs and send them to UVic, where technicians look at the species inside the stomach. This clever program provides us with a way to look at trends in space and time at different forage fish such as sand lance, northern anchovy, and herring, which are all vital for Pacific salmon as diet components. Another major group of projects that we carry out under the Marine Science Program fall under the Nearshore and Estuary Research and Restoration umbrella. These projects aim to restore nearshore habitats, improve coastal function, and carry out research projects that will help us determine restoration methods that will be most effective and adaptive under climate change. Shown right here is an example of a project that we funded to develop an eco-friendly mooring system. This allows a mooring line to be suspended at low tide instead of laying down on the seabed, and this results in less damage to the seafloor and protects eelgrass and kelp beds from scour. These are both vital nursery habitats for Pacific salmon. Another program that we are developing is to create awareness of the impacts of climate change to coastal areas and tools that citizens can use to make a difference. Traditional methods of protecting coastal communities have been the use of engineered or hard structural solutions such as seawalls and riprap, breakwaters. And these structures might protect the single property that they are built on, but they interfere with shoreline processes, which results in increased erosion elsewhere, loss of beach habitat, and flooding, which can often happen down the beach for your neighbors' properties. And of course, the loss of these beaches has major impacts on forage fish habitat. So in recent times, there's been a move to these softer, nature-based approaches such as green shores. And these approaches increase resilience to climate change, and the associated plantings that take place at the top of the beach and also in the riparian areas behind them helps to sequester carbon. So these are good for combating climate change. We are also partnering with DFO to create awareness of the risk of aquatic invasive species such as green crab, which can cause major damage to eelgrass beds. And here is an example of a sign that we have created at PSF, as well as many other outreach that we have worked to provide, such as flyers and rat cards, which have been distributed throughout BC. We also understand the importance of removing debris, as this hinders successful restoration of nearshore areas. So we support work to remove garbage and debris that is collected in nearshore areas and on the beaches themselves. Moving forward, we're hoping to map the locations of creosote pilings throughout the Strait of Georgia. Herring often lay their eggs on these, and these eggs end up dying because of the toxic nature of creosote. So we're hoping to work with partners to remove these toxic structures from our waters. We also have many other programs in the nearshore category. One is with our partners at UVic, which is including the identification of resilient strains of kelp and understanding why these persist. These particular strains may be really useful for restoration and will likely be the ones that we will identify for protection and storage in a new biodiversity bank for macroalgae that we are developing with partners at SFU. This will be a similar concept to the Doomsday Seed Banks, but will be for marine plants. And we're also working on a major climate adaptation plan for nearshore habitat restoration. Another major program that we have in the Marine Science Program is our Bottlenecks to Survival program, which is basically a large-scale extension of the work we did in the Cowichan during the Salish Sea Marine Survival Project. And here is Colin Middleton. He is a new hire at PSF, and he's the program manager for the Bottlenecks project. He's injecting a small pit tag into a juvenile salmon. This major program is funded by the BC Salmon Restoration and Innovation Fund and aims to determine the survival bottlenecks in freshwater and marine environments for Coho, Chinook, and steelhead in southern BC, focusing also on the winter period, which is identified throughout our SSMSP program as a really critical period of the life history of salmon. And for this program, we are partnering with the BC Conservation Foundation as our major partners, also the province, DFO, UVic, and many First Nations and stewardship groups around the Strait.
Hey there, I'm Colin Middleton. I'm a biologist with the Pacific Salmon Foundation, and we're here this morning with the BC Conservation Foundation and staff here at the Nanaimo River Hatchery, and we're tagging juvenile coho salmon with pit tags. Let's go check it out. So this is a pit tag, small little tag about the size of a grain of rice. It has an individual identification code associated with it that allows us to track these fish through different points in their life history as they migrate out through the freshwater and into the marine environment, and then again as they return as adults. We'll use this information for survival estimates at those different points. And this is part of a broader study being put together by PSF and BCCF looking for survival bottlenecks throughout the Salish Sea.
The Strait of Georgia Data Center, a collaborative program between UBC and PSF, is where we store all the data from our programs, as well as many other external programs, community groups, First Nations, and academia. The data center provides open access to marine ecosystem information, including human use, ecology, and environmental data. We're currently working on a Strait of Georgia Marine Reference Guide where many of these data sets will be visualized as map overlays. The layers shown on this side were plotted to try and help our bottlenecks team determine the best places in the Strait of Georgia to catch juvenile fish over their first summer and winter of life. Another BC SRIF funded major project that we are carrying out is a review of hatchery effectiveness. As Brian noted, our studies in Cowichan showed that hatchery fish appeared to survive much more poorly than wild fish. This study aims to review cutting-edge molecular tools and research that might be applied to improve productivity of hatchery-reared salmon, to evaluate different hatchery release strategies to see if some are more beneficial than others, and to examine wild-hatchery interactions. Another program is focused on how to address questions around predation. We've been funding some further research since SSMSP on the extent of predation on adult Chinook returning to the Cowichan River. And it seems that river flows are crucial for both juveniles and adult Chinook. Low flows result in higher predation of juveniles, particularly by herons as well as seals. Herons in particular like to forage in shallow water, so as soon as flows are low, these small juvenile fish become much more accessible. And at the same time, when we have low flows when adults are returning to these systems, it delays their entry and also results in higher predation by seals. And log booms really appear to exacerbate the issue. Both juvenile and adult fish hide under these and become sitting ducks to seals, which use these log booms as haulouts. Also, when we have a very high tide, seals are not washed off the log booms, which they often are from their rocky haulouts. And this, therefore, keeps them protected from transient killer whales, which would otherwise feed on them and prey on them. So now we are looking closely at both the importance of implementing minimum ecological flows, as well as trying to get log booms out of estuaries during these critical salmon migration periods. Finally, we're working to create a number of different outreach products that will be accessible to all different groups, ages, and people throughout BC. These are focused on stewardship groups, the general public, government, and also more recently we've started creating more educational products for the K to 12 age group, based very much on our SSMSP findings. So please do check out our PSF website for educational videos, games, and activities for children. The focus of these programs is really on making our science from SSMSP and all of our existing programs accessible to everyone. And this, I think, is a really important goal moving forward. Clearly, we could not have done any of this research without the integrated and highly collaborative groups that we work with, both in Canada as well as in the US. And we also could not do what we do without the support of you all, our donors. So moving forward, we are really hoping to sustain the networks of researchers, communities, citizen scientists, governments, First Nations, and academics that were built up and created over the last many years for PSF, and trying to continue to put our heads together to make a difference to salmon. Thank you, everyone.
Thanks, Isabel. It's wonderful to see all the development since the SSMSP is finishing up and where we're going in the future. But it does lead to some bigger questions for people on how do we sustain this collaboration? How do we sustain our role in the Salish Sea? I'm going to present just 3 ideas to you here, and I call these the bigger ideas. Number 1, I think it's critical to sustain collaborations and to protect salmon. I'm suggesting that we might consider an international Salish Sea Council, including government, academic, corporate, and community reps, and a regular publication of a State of the Salish Sea report that could be on various topics of interest publicly. So that's just number 1. Number 2, there are so many positive things that we could do to repair the habitats and improve conditions for salmon. I'm suggesting that we create a habitat restoration fund to support salmon conservation in the Canadian Salish Sea and to support climate change action under the federal blue economy. And that'll nicely integrate with PSF new climate change for salmon studies. And thirdly, I think we should find a way to create a BC Salmon Corps to educate youth, employ youth in environmental restoration and climate action, and to promote cross-cultural exchange of youth. That's it for me. I take great pleasure in having managed this program for you for, almost a decade. So that's time to finish up our presentation. Let's pass it back to Jason to manage a moderated question and answer period. Thanks, everybody. It's been great.
Well, thanks for that great presentation from Brian and Isabel. And it's my pleasure now to turn to some of the questions that we have. And Corey is queuing up questions on the Slido. But to get this started, I'm going to ask a question of Isabel and Brian around the Salish Sea Marine Survival Program. And what were some of the most interesting or surprising things that you learned from the studies in the SSMSP?
Ladies first, of course, right?
Sure, I'll go ahead. I think maybe Brian had mentioned a little bit of this, but when we were doing the pit tag program, we found that 4 years, which were 2015, 2016 in particular, we had this very weird, big, warm conditions, very low flows. It wasn't just that, fish, but actually 70% of the fish died on their route down the river.
Hey, so, Isabel, can you hear me?
Are you getting feedback? Because I am.
Yeah, we're picking up a bit of feedback or something. I'm not sure if...
I'm just talking into my computer. Oh, that's better. Okay, great.
Thanks.
Yeah, I wasn't sure if you could hear it. So when they were doing the pit tag studies, the hatchery staff would release the fish right at the top of the Cowichan River, and the assumption for this indicator stock was that there was 0 loss downriver. So I think that was pretty surprising that actually in those hot, low flow years, you could lose 70% of the fish. And then obviously the question was, what is happening and what are the reasons for this? And so, they put down a lot of trail cams, and they had a bunch of pit tag arrays on some of the channels and along the river. And then the unusual finding when they did that, I don't know if you have the image that I had provided that you can show to people, but this is what they were finding on the trail cams. Now the black and white picture's a little hard to see on the left, but basically they had been seeing pit tags moving upriver. And so they're scratching their heads like, how could these fish be moving the wrong direction? And it turned out from the trail cams that obviously these pit tags were in the bellies of families of raccoons that were able to forage and pick them off as they came downriver. And as they walked up, they would cross another array, and it looked like pretty strange behavior. And the second surprising thing was just how many pit tags were found underneath the heron rookery in Cowichan, where we have about 100 nests. And they found hundreds and hundreds of pit tags, and most of the tags that they picked up from the 2015, 2016, and 2017 years of study were from 2016 when flows were particularly low. And so herons seem to be very good at picking off, young fish from the estuary and lower river. They prefer to forage in really shallow water, so it made sense they got more in those low flow years. But interestingly, over 70% of the tags that were found under the rookery had come from hatchery fish as compared to wild. And these studies were comparing the losses of both hatchery and wild. So that was pretty interesting too. Obviously, the hatchery fish are bigger and probably a nicer item to eat, but definitely had a lot more impact in terms of losses. So I thought that was pretty interesting. I think Brian had something to add, did you? Over to you.
Yeah, I think there were probably a few that would jump to mind for me. I think the first one that comes to mind is right in the very beginning of the program when we started thinking about how to deal with predation. I'm sorry, a PhD student from UBC just say, well, why don't we just measure the mortality directly, right? And that led to the seal beanies. It was a fabulous start for the program, and it worked spectacularly. I think we're all shocked, or surprised at least, at the magnitude overall that's estimated. So we're going to continue to deal with that one. Probably one that we haven't spent a lot of time on is, I would have to say that the success of the Strategic Salmon Health Initiative with Dr. Miller and students and Dr. Emilio DeSico. We just keep finding things. The more work that we do, the more we find, and now we're actually going into studying the sea lice that we hear about, but they carry a whole different suite of viruses.
Okay.
So there's all sorts of things that are surprises, to be perfectly honest.
All right, well, thanks, Brian and Isabel, for that. And that was my moderator's prerogative to ask the first question. And now I'll turn over to Corey to see what we've got coming on the Slido.
Right on, yeah. We've got a few coming in on the Slido, but keep them coming there. And we had a few questions ahead of time from people as they were RSVPing, including one from Tony Allard. And his question is, on the heels of FOPO, or the Standing Committee on Fisheries and Oceans, that was earlier this week, will the work of the Strategic Salmon Health Initiative continue?
Thanks, Tony, and thanks for your continued support over the years. Absolutely. We've already had this discussion internally. The directors have agreed that we should invest in continuing work to follow up on completion, hiring Andrew Bateman to come in as a strong spokesperson about the work on the pathogens. He has excellent studies in epidemiology and modeling, so we've got a strong person coming in. That's a big commitment. We have sustained Emiliano ongoing. He's working with the Namgish peoples in the Broughton Archipelago and working to develop what's called eDNA as a way of very closely monitoring the farms if they're populated in the area. And we have other studies that are in discussion right now that are continuing, particularly about the means now that we can do gene sequencing so we can identify different lines of a pathogen, or whether it's a virus or a bacterium and so on, so we can actually trace how the animal is evolving in our waters. And there's ongoing studies about the sea lice, as I already alluded to. So yes, there's going to be a very active program, and it's basically funded over the next 2 to 3 years at this point.
Great. Any follow-ups there from anyone? Isabel, or no? We're good?
Yeah,
all right.
we're good.
We got lots more in the Slido here, so we'll move on. The next one from Dan Nomura, and this was actually, he asked these questions during our last major donor event, but we've got a few other herring questions already, so we're going to kind of lump them together and ask, what was the role of the SSMSP as it relates to herring, just generally?
Isabel, do you want to start that one with DFO? Yeah.
Sure, I can make a start. So we did assist in funding the Young of Year Herring Survey, which was undergoing some funding constraints during SSMSP. So that was continued each year and provided a huge amount of information. And obviously, there were really focused studies on herring on both sides of the border, just concerns about the herring and how the role that it plays for both Coho and Chinook, particularly over the first summer when they are switching over to feeding on fish. And so a lot of diet information and gathering of general information. And in terms of some of those studies, we've also done a lot of mapping and work to look at the changes in herring spawn distribution And some of the work that was done in the Cowichan was really focused on the interplay between, availability of herring and also the risk of predation on a very local scale. And when we were comparing from, the 70s to this current work during the Salish Sea project, it was apparent that herring was probably at a higher level, in proportion of the diet in the 70s, and also available over a longer period of time. So, it seems that there have been some reductions in the amount of herring in the diet, but the fish, the Chinook that were caught and that had been feeding on herring were the bigger Chinook, and they were also growing much more quickly. And it seems to be possibly a bit of a mismatch, with us maybe losing diversity of spawning of herring. We don't have availability for as long a period. So in the spring, in the 70s, Chinook would have been feeding on herring. Now they're not. The little herring are not available. And so there appeared to be a little bit of a mismatch that really only the biggest Chinook were able to feed on herring, and that had, impacts on their growth and survival. And those smaller Chinook would be feeding on, zooplankton over the winter, which disappears. So who knows what happens to them. I could carry on a lot on the herring question, but maybe I'll just pop over to Brian and see if he has something to add right now.
I think the most important thing to add, actually, Isabel, is if I was identifying key gaps where people should really try to fill some of this in, I mentioned the bird predation in total, but I think that the population, the dynamics of production of herring in the Strait of Georgia, the very early stages of development, is one that is not particularly well studied or understood as yet. We picked them up in September with the juvenile sampling surveys, and the abundance we see there used to be very closely aligned with the survivorship we saw of the choco sockeye salmon. But that has broken down and has become much more variable and more modest in terms of abundance. So it wasn't a major component of the Salish Sea Marine Survival Program. If I was to pick something to really focus on as a new PhD, as I wish, then I would say I'd be looking at the early dynamics of Pacific herring in the Strait.
All right, very interesting.
Okay.
The next one's from Anonymous, so I don't know who asked this one, but what are you finding and what's needed in the eelgrass/estuary areas related to natural climate solutions and the obvious nurseries for salmon and forage fish? Yeah.
I think that's an Isabel one.
What are we finding? Sorry, I have to just read that one.
And what's needed?
So in terms of, I still have that echo, which is strange. For the climate solutions, you still have the echo?
No, we're okay on this end.
Oh, strange. It's just coming back to me. Some of the things that we're trying to focus on are firstly identifying thermo-tolerant species that could be most useful for restoration. So that is one of the things that we're doing. A lot of the restoration work that is ongoing, is quite successful, but it's going to get harder and harder with warmer waters. So I think finding the right species and matching those is really what we're focusing on in the type of research that we're doing. The biobank will aim to, make sure that we are storing genetic material for when we do find that we're needing to restore an area, that we don't lose these species before it's too late. And a lot of the other work around sort of the soft shores and the beach nourishment and the work that we're doing to try to, improve conditions and shoreline processes will tie in and make it a lot easier for, not leading to major issues with sea level rise and storm, just destroying these nearshore habitats completely. And the other part of that is just, thinking ahead and working with other groups that are looking at how to even create resilience in these regions. And one of the most important things is, of course, having room to sort of move up as sea level happens, and trying to make sure that you have good connectivity for all of the different habitats, marsh and eelgrass and kelp, so that your fish have escape habitat throughout these different locations.
Just something to quickly add. is some of the work going on in Puget Sound that we've been able to be part of a little bit has really showed that you can restore very productive estuary type habitat very quickly. In major programs in Nooksack and Skagit, where they've taken estuaries that had been used agriculturally for decades, they turned it back into connected estuary habitat. And in 2 to 3 years, you couldn't tell the habitat from the pure estuaries in terms of food composition and species and so on. So they're incredibly resilient to restoring them. And lots of community effort could be put into replanting sedges and eelgrasses and then working with nearshore kelps. So I think there's a great opportunity to do work there, particularly even under climate change programs.
Well, that's great. Just a quick follow-up, actually. Isabel, for you, we've shown a lot of the work about the eelgrass restoration projects in some of our past publications. Is any of that work going to be continuing on?
The restoration work itself?
With Nikki Wright's group? Yeah, the actual restoration work.
Yeah, no, absolutely. We continue to support lots of groups, including Nikki and her restoration work. So yes,
great.
The short answer.
Okay, perfect. We've got a lot more on the Slido here. So next up is one that we receive very, very often, and it has to do with predation, namely on salmon by seals. So what are the next steps with regard to predation of salmon by seals?
I can make a start on that one. So we had discussed during the talk a few of the sort of immediate thoughts around what we can do. And, the thing to remember is that juvenile salmon are actually a very small, very tiny, in some years, proportion of the diet of seals, maybe only 3%. And most of the diet is of herring and hake. So I think a lot of the ways that we're looking at this is, how have we created impacts to the environment that make seals more effective, predating on salmon, and put those salmon at much more risk. And so we're trying to start with, what are the things that we can do to make habitats safer for salmon. So I don't know, I've got a slide or two for this because I thought it'd be interesting to talk about some of these different aspects. I don't know if you can pull those up, Corey. Yeah, so you got that? One of the things that we're looking at are that over time, hatcheries have been releasing their fish in a much more contracted period of time. So this is just a graph just showing the changes in releases in the Strait of Georgia and Puget Sound over time. And you can see that from around, just before 2010, we've been pumping out millions of hatchery fish into the Salish Sea, basically in a 2-week period at the end of May. And so this is really kind of like converged, this timing, and that means that suddenly you've got a huge number of salmon, baby salmon in the Strait, which in itself could lead to kind of switching, by seals to those salmon. Whereas if this was a much more diverse strategy and these releases were taking place over a longer period of time, it probably wouldn't be, worth the while of the seals to make as much effort to feed on these. So this is one of the things that we're looking at is, in our Hatchery BC Thrift program, and looking at whether a sort of more diverse suite of strategies around release timing might be more effective. Instead of these huge pulses, should we also be trickling fish out instead of them coming out in a big lump? And then I had another picture there, I think, that I wanted to chat to you about, which is that seals, as I said, mostly focus on herring and hake. And one of the things that was found during the Salish Sea project is that when there are other forage fish that are abundant, sometimes we may get something where there's a switching to those and we get less predation on salmon. And this is just the graph showing survival through Puget Sound of young steelhead that are coming out. And in those years with the red dots, which is when there's very low anchovy abundance, that's when you're getting the lowest survival rates. And then the years when you've got the green dots, there's a lot more northern anchovy around in the Strait. And those years coincided with higher survival. So it seems like when you've got other forage fish that are very abundant, that the seals will cue in more on those and hopefully save our steelhead and salmon. So I think this leads to a lot of the work that we're trying to do to make the environment better for other forage fish. So some of the considerations in this restoration work is to make better habitat for herring, to look for different ways to increase different types of forage fish. So we also want to think about sand lance and other species like that. We want to create more escape habitat for the salmon when they come out, so they actually have somewhere to hide. And of course, getting rid of log booms and looking at pinch points and places where the fish are most at risk, I think will also be really important. And the final picture that I had in that little slide deck, which you probably have, is just a reminder that, the log booms are one issue because they're a nice little platform for the seals, and whether it's juvenile or adult salmon, they're sitting ducks when they go and hide under those log booms when they're floating, and the seals can just hop down and feed on them. But other issues with the log booms are obviously sort of loss of logs onto our coastlines, which we've all seen on many of the beaches around the Strait. And these logs can create a lot of erosion and scour, but they can also completely clog up a lot of the beaches that are important for forage fish. So if you've got a great beach for sand lance or surf smelt, and there's no way that, any fish or, sand lance can come and spawn on that beach because of the coverage like this, that becomes a problem too. So I think it's sort of a multi-pronged kind of approach, looking at the issue of flows, which was mentioned before, water temperatures. We need to look at these in river so that fish can actually enter the river before they're at risk or leave in good shape when they're young. And then, making the habitat as hospitable as possible for all forage fish, as well as escape habitat for salmon. And again, that whole connectivity piece is really important so that the fish can kind of come out and move from one of these sort of, key marine nearshore habitats to the next one without becoming exposed to high levels of predation. And the most important part of this, I think, is really thinking about the herring question, because with the loss of some of those southern Strait of Georgia spawning areas, it's probably putting a lot of juvenile salmon under a lot more risk because they're having to move further to look for herring to feed on. And when they're doing that, they become more exposed to seals. So I think the reestablishment or looking at ways to really promote more expansive spawning areas for herring is really important too.
Well, great.
Brian, do you have
Brian, did you want to jump in, or did Isabel answer that to your satisfaction? I think he's good. It's obviously a pretty complex issue for sure, and Isabel, I can attest to some of the increased anchovy populations in the Strait just in the last few years. I know a lot of the fishermen are seeing just huge balls of anchovies out there, so it's great to see that some of those forage fish are coming back in good numbers. We're going to move along unless, Jason, you had anything to...
No, let's stick with the Slido.
We'll stick with the Slido here. So next up, we got a question from our good friend Terry Tebb. How you doing, Terry? He says, congrats on this very valuable and much needed work. Does DFO actively support this ongoing work with technical support and dollars?
I think the...
Go ahead, Brian.
Go ahead, Jason, if you want to.
Well, I was just going to start with the BC SRIF, the BC Salmon Restoration Innovation Fund, which has been a substantial source of funding for some of the work that Isabel and Brian have described today. The hatchery effectiveness study that Isabel and Brian are leading is funded by that as one example. So additionally, there's a project related to nearshore habitat that Isabel just successfully wrote a grant for that's being funded by Environment and Climate Change Canada. So that's just a couple of examples that I thought I'd start with. And Brian, I'll turn it over to you.
No, those are good examples. I think the other element is they are continuing the Georgia Strait trawl sampling on an ongoing basis. So that's a significant contribution. They do the DNA analyses. So I think overall, Terry, the answer is probably yes. There have been some more difficult working relationships related to our discoveries in the Strategic Salmon Health Initiative, but I think that these have all been, over the long run, have improved the science. We're very, very careful and methodical about how we proceed in our programs and that. And that's just to ensure that we're doing the best science for the money we were investing. So I would say it's not bad overall, and it's the most recent budget. And great to hear from you, Bob.
Okay, next up we got a question from Alan Wiggin. I think he's over in Salt Spring. To aid management, any plans to assess relative, it moved on me, sorry. Any plans to assess relative priorities of diverse factors studied, considering impact on production, timeliness, cost, and practicality? I hope I read that right. You guys get that?
Yeah, I'm just trying to think of what specific question here. I mean, I think as we start talking more generally, Alan, about big restoration investments that you heard about in the recent budget, we are thinking about how you tie all these programs together, and how do you make the best benefit for salmon from a number of smaller investments. The timeliness, cost, some of this gets down to how much you're prepared to invest in trying new things. A really simple example that we've touched on a little bit, and one of the things that we think should be done with Pacific herring is to try and restore some of the diversity of lost spawning areas. But I think those areas have maybe been lost because of marine plants and temperatures. And so a very simple thing we could try is allowing spawn-on kelp in certain areas with the herring and then to move the spawn. Be a very slow process, but it's very doable. And so, yes, we look at cost and practicality, but basically we want to start by focusing on where are the greatest needs and what can we assist with. I'm not sure I can give you a much more specific answer, to be honest.
Well, I could maybe just link that question around relative priorities of diverse factors to another initiative that we've been pursuing that Mike Menier has been leading us on, and that has been reaching out to our neighbors down in Washington State who seem to be fairly advanced in some of their thinking around how they assess priorities in the different watersheds and regions for salmon, and then start to organize those so that there is some structure to how they prioritize, how they resource their discretionary funds towards those priorities. So through Mike, we've managed to connect with senior people in the provincial government, people in the Washington State Governor's office, and it seems like there is some good thinking that has been occurring in Washington on their salmon planning that we might be able to use here in BC towards organizing and prioritizing some of the issues here. So there is a cross connection there between some of the work that has been led under the Salish Sea program and some of these issues around where do we go from here.
All right. Well, great. We've just got a couple minutes left here, and I think we'll end on one that is donor-related, since that appeals to my heart. So the question is from Anonymous, but it says, you've offered 3 strategic ideas, I think those were your big ideas at the end there, Brian, to carry SSMSP moving forward. And yet, what are you most worried about that us donors can help with? It's a great question.
Thanks. That is a good question. And I think you can help with all 3. Because they will take some pushing. Probably the one that's the biggest help with the donor is I think we need to be more innovative about how we build a restoration fund. We've come up with many, many activities that can be done to improve our knowledge and to restore restore habitats, support small community hatcheries in different ways. And I think the problem is going to be there's going to be far more need than there is available funds if we have to rely on government only. And then if you do that, you have to follow the processes. The BC Salmon Restoration Innovation Fund is great to have it available, but it's a relatively slow process, so you don't have any guarantee that you could build a major program through it. So having money available through a restoration fund is a wonderful way to go, so you can prioritize the work. You can do it independently, work with all the various community groups, and that I would say thinking creatively about how to build that fund so we have money to invest in all these good ideas probably the biggest benefit we could possibly get from all you guys that have great ideas.
All right. Well, thanks very much. I think we were probably out of questions. So, Jason, I'll turn it back to you if you want to close things out.
All right. Well, I'd like to say thanks again to Brian and Isabel for your presentations this evening. It's really been my pleasure to moderate the session, and I've enjoyed hearing the story about how the SSMSP went from an idea to becoming an international initiative involving hundreds of people. I'm really pleased to know and be part of the PSF initiative to continue and carry this work forward, working on new and exciting projects for our salmon and their ecosystem. Great work like this all starts with great people, and PSF is very fortunate to have Brian and Isabel on our team leading this work. So I'm now going to turn it back to Corey for some closing remarks.
Well, thanks very much, Jason, and of course, Brian and Isabel and Christina McIntyre, who put this whole thing together behind the scenes. And thanks, of course, to all of you for joining us tonight, for participating in the discussion. As we mentioned earlier, if you did ask a question that we weren't able to get to during the live Q&A part, or if you've got a burning question you'd like to ask now, we will have the Slido open for a few more hours here, so feel free to get your questions in, and we'll make sure we follow up with you in the coming days. Many of you tuning in tonight are some of PSF's most prominent donors, helping to fund programs in salmon science, conservation, and restoration. And now, more than ever, we're so grateful for your support. As a reminder, thanks to the support of the Pacific Salmon Endowment Fund Society, as well as that of the federal and provincial governments, 90 cents from every dollar donated to PSF goes directly to programs like the one we just learned more about here tonight. And as we look to the future, it's the support from you that will enable the next steps of this research, as well as many other important initiatives for salmon that PSF's currently undertaking. So please stay connected, please keep in touch, and by all means, please introduce us to new folks who might be interested in hearing more about PSF. Finally, we look forward to the time, and hopefully it's very, very soon, where we can all gather again, because we do really miss seeing you all in person. And with that, I'll say stay safe, stay well, and good night, everybody.